In this video, we are going to be talking about our last biomolecule, which is lipids. When it comes to lipids, let's understand that lipids can be divided into two separate families, which are, as you can see on the left, complex lipids, which can be hydrolyzed, and the molecules that we have on the right, which are simple lipids, which those are lipids that cannot be hydrolyzed. Now, what is a lipid? Lipids, the word actually comes from the word lipos, which is a Greek word, and lipos literally means fat. So whenever we're talking about lipids, understand that we're talking about fat. Now, when it comes to complex lipids, that includes the molecules that are known as waxes, triglycerides, and another word for triglycerides is triacyl glycerols, and lastly, phospholipids or phosphoglycerolipids. One of the things that it is very important to notice when it comes to complex lipids is that if we actually focus on the functional groups that are present in these molecules, it doesn't matter if it's waxes, triglycerides, or phospholipids, we are going to have in many of these molecules an ester bond. There could be one in the case of waxes, there can be three in the triglycerides, or there can be two, like we see in the phospholipids or phosphoglycerolipids. Now, when it comes to the simple lipids, the ones that cannot be hydrolyzed, understand that we have three different members of that family, which include steroids, prostaglandins, and terpenes. Now, understand that when it comes to the simple lipids and what it means that they cannot be hydrolyzed is just that these molecules are going to be building blocks and are going to be utilized further into the synthesis, or they're going to be involved in some communication systems, but they are not going to be breaking into pieces just like complex lipids are. Now, when it comes to lipids, understand that as you can observe, there's a variety of structures that are present. The only common feature that all lipids have is that they are soluble in organic solvents, but not in water, which means that most lipids are going to be non-polar molecules. Let's first talk about the family of complex lipids, which is waxes. So this is the first member of that group. So when it comes to waxes, understand that waxes are high molar mass esters. And when it comes to this, understand that there's going to be two components of a wax. As you can see, you're going to have a component that it is to the left, and that is going to come from a fatty acid. And that fatty acid is going to be, in essence, a carboxylic acid. And that carboxylic acid is going to be linked to a long chain alcohol. And this long chain alcohol, as you can see, is just going to have many carbon atoms overall. So because we have an ester functionality present in waxes, then you can envision that hydrolysis of a wax is going to produce the two components. Again, when it comes to biochemistry, this is just application of knowledge from previous chapters in terms of functional groups. We learned previously that esters hydrolyze to carboxylic acids, and let me put it as singular, and an alcohol. So it is not surprising to know that since we have one ester bond in a wax, then the hydrolysis of them are going to produce those two functional groups that were just described in the second bullet point. Now, understand that waxes can only differ in length of the carbon atom chains on either side of the ester, and then they serve a variety of functions in living systems. Mainly, one of the things that waxes are used for is for retention of water, for example, in plants. Now, understand that waxes can come from animal sources or they can actually come from plant sources. The second member out of the family of complex lipids is triglycerides. And as I mentioned previously, they are also known as triacylglycerols. And the reason why they know as triacylglycerols is because, as you can see, we have three acyl groups bonded to the glycerol unit, which is going to be the alcohol portion. That has to bind together to these fatty acids that you see boxed in green in order to produce it. Now, understand that triglycerides are triesters, as I previously mentioned. So there's three ester bonds, and as we, remember the reactions that we have seen previously, those three ester bonds come from the idea that when you have glycerol and you combine it with three fatty acids, in essence, what we're doing is that we are combining alcohol motifs with carboxylic acids. And as we learned in the carboxylic acid chapter, when we combine an alcohol and a carboxylic acid, esters is what is produced. Now understand that triglycerides are generally used for long-term energy storage. So we discussed in a previous video that we store sugar in the form of glycogen. We also store fat. We don't store amino acids. That's why it wasn't mentioned previously. Amino acids, we use them for energy. They make the proteins, but we don't have an actual storage of amino acids that is specifically utilized for energy. Now, understand that if we calculate the amount of energy that burning fat actually produces, burning fat because you are processing these type of biomolecules through a metabolic pathway in order to produce energy in the body, you can see that fat is going to produce 9 kilocalories per gram versus carbohydrates and proteins only produce 4 kilocalories per gram. And again, these metabolic pathways of how we produce this energy is not discussed in this course. I'm just trying to show the commentary that maybe some of you have heard, that when you burn fat, that produces more energy than when you are burning carbohydrates or proteins. Now, when it comes to the triglycerides, we know that fatty acids is one of the components that are present in there. Now, fatty acids can be from 12 to 20 carbons in length. They can be saturated or unsaturated. So what makes a saturated fatty acid is going to be the presence of carbon-carbon and carbon-hydrogen single bonds in the tail of a particular fatty acid. What makes something an unsaturated fatty acid is going to be the presence of one or more carbon-carbon double bonds in the tail of that fatty acid. As you can see here, stearic acid is an example of a saturated fatty acid, and oleic acid is an example of an unsaturated fatty acid. When it comes to shape, specifically for fatty acids, understand that fatty acids, depending on the structure that they have, we see how that affects their melting point, for example, one of those physical properties. So let's say that we are comparing specifically a saturated versus an unsaturated fatty acid. And I understand that they have different carbons in length, but even if we're comparing things that have the same number of carbons, these trends remain to be true. So understand that saturated fatty acids, as you can see in the numbers for the melting points, when we compare them, have higher melting points when compared to unsaturated fatty acids. And you may be wondering why that is. The reason why saturated fatty acids have a higher melting point than unsaturated fatty acids, one of the ways that we can envision it is that they have higher molecular weights, okay? And understand that when it comes to this, mainly we have to think about attractive forces. Okay, so these trends that we see in the melting point have to do with the attractive forces that are present in these molecules. Now, when we look at these molecules, even though they have a polar side to them because of the carboxylic acid, the fatty acids are overall nonpolar molecules, which means that the strongest attractive force that they have is specifically dispersion forces. So, how does this influence the melting point? Well, this is what occurs. In saturated fatty acids, one of the key features of these dispersion forces is that because we don't have that carbon-carbon double bond and it's just carbon-carbon single bonds, saturated fatty acids can stack. So, because this saturated system can stack onto one another, they're going to have higher dispersion interactions. To utilize an analogy, it's kind of like if you had potato chips and the saturated fatty acids will be kind of like having Pringles potato chips, in which each of the chips are going to stack onto one another. That is going to have a greater dispersion force interaction compared to the unsaturated. Because what happens in the unsaturated system is that having this carbon-carbon double bond creates a kink in the system, and this kink, quote unquote, what creates in the molecules when they're interacting with one another is like they do not stack well. And since they don't stack well, they're going to have reduced dispersion forces. And overall, we see these effects in the melting point. So you can visualize, to utilize an analogy, that when it comes to unsaturated fatty acids specifically, it's kind of like having Lay's potato chips, that even if you try to make a stack, they will not stack as high as something like Pringle potato chips. So understand that this increased interaction of saturated fats is what results in these observations that we have for the melting point. Now, understand that these effects of the fatty acids translate into what we observe for the triacylglycerols, because triacylglycerols or triglycerides are going to have those fatty acids present in them, specifically 3 of them. Now, it is known that when it comes to looking at different triglycerides, we can divide them into fats and oils, okay? So triglycerides that are fats are going to be solid at room temperature. And these fats are going to come from animal sources. Oils, on the other hand, are going to be triglycerides that are liquid, and they're mostly come from plant sources. Now, if we look at fats and oils specifically at the structural level, these molecules that we see here, which are the balls, and stick models, molecular models for them, they are just going to be illustrating what do we see overall in fats and oils. Understand that when it comes to a fat, we are mostly going to see, as you can see, these straight chains here, which means that fats are going to have saturated fatty acids. When it comes to the oils, they are going to have unsaturated fatty acids. And additionally, as I just mentioned, the fats are known to come from animal sources. Oils tend to come from plant sources. So that's why, like, for example, there are some plant sources that have saturated fatty acids. So coconut oil has saturated fatty acids. So for those of you that may have coconut oil at home, you know that it is solid at room temperature. But that comes from the idea that the fatty acid that forms coconut oil is saturated, so that's why it's solid at room temperature. It is not considered a fat because it is actually coming from a plant source, so that's why we call it coconut oil. Now let's mention some of the reactions that are involved in triglycerides. The first one is hydrogenation. When it comes to hydrogenation, specifically of triglycerides, you can observe that if you have a particular triglyceride, one of the things that I should mention before I continue is that when it comes to the unsaturated fatty acids, if you haven't noticed before, in nature, unsaturated fatty acids are cis in configuration. You can observe it here, just in case if I forgot to mention it before. But going back to hydrogenation conditions, when hydrogenation conditions are occurring on a triglyceride, understand that this is going to be the conversion of a alkene to an alkane. So as a result of the hydrogenation of a triglyceride, we are going to have a saturated triglyceride. Now, when it comes to this, understand that you may be wondering, why are people actually doing hydrogenation on triglycerides? Well, hydrogenation of triglyceride actually, it is produced in the food industry. So for those of you that consume margarine or shortening, Crisco, if you don't know what shortening is, those products actually come from the hydrogenation of plant sources. So whenever you buy, let's say, a margarine that comes from olive oil, what they did is that they took that olive oil, and that the olive oil has fatty acids that are unsaturated, and upon hydrogenation, then you are creating now a product that it is partially hydrogenated. Now, understand that partial hydrogenation, okay, is what gives rise to the products that I described previously. And as you can see, it is noted in the first bullet point. But understand that one of the interesting things about this process is that during partial hydrogenation, that one of the products or byproducts that can happen is that it can produce trans fats. So the problem with trans fats is that we don't have the biomolecules to actually deal with them, and that's why they are so bad for you. So understand that the creation of trans fat, specifically during hydrogenation, this is a byproduct, is not the main product of the reaction. The main product of the reaction is creating that saturated fat. Now, understand that in the production of this trans fat, you may have observed, if you have these products at home, that all of a sudden, if you buy, margarine is a great example, that when you buy it, it actually says partially hydrogenated oil with zero trans fat, because what happens is that the food industry, for that product, they remove all the byproduct trans fat that was created. So if you are doing a predictive product in hydrogenation or triglyceride, please just place the saturated fat as a product. You do not need to include the trans fat byproduct. Hydrolysis is another reaction that triglycerides can undergo. So hydrolysis, as you can see, it can occur in a vessel, meaning outside of the body, and that means that if you place a triglyceride in aqueous base, what you're going to be forming is specifically the alcohol. So if we think about it, this is an ester, and if we put it in base, this base hydrolysis of a triglyceride is called saponification. And in fact, saponification, which is the base hydrolysis, we are going to produce an alcohol, and we are going to produce carboxylate ions. Now, this reaction you have observed previously, because this is the base catalyzed ester hydrolysis. It's basically a nucleophilic acyl substitution for an ester. But understand that this reaction actually is important because those carboxylate ions that are produced through the saponification reaction is what makes soap. Now, when it comes to the formation of this soap, understand that the reason why these molecules can work and make soap is that now that we have those carboxylate ions, these molecules are actually soluble in water because it has an ionic component. And when we think about how soaps work, pretty much they're going to be interacting with the soil that you're trying to remove from clothes or from food. It doesn't matter the soap that you're using, if it's dishwasher, if it's body wash, if it's laundry detergent. All soaps are going to work in the same way, in which the non-polar tail that it is produced, or that it is part of the carboxylate ion, is going to be interact with the soil portion, and then this part of the molecule, which is the ionic interaction, that part is what's going to interact with water. And what I mean with soil is whatever the soap is trying to remove, body oil, food particles, so on and so forth. Now, the main point that I want you guys to take out of this slide is that when you have an ester and you saponify it, you're going to make an alcohol in the form of glycerol and carboxylate ions.